Welcome to the screencast on animal cloning. I'll start off this screencast with a slide I also showed at the start of my screencast on cloning in plants, just showing the difference between two ideas of cloning: reproductive cloning and non-reproductive cloning. Reproductive cloning is cloning to create new individuals. We looked at this in terms of plants, in terms of our micropropagation and vegetative propagation, would be examples of reproductive cloning. We'll look at that with respect to animals. Whereas non-reproductive cloning is producing cloned cells which can be used or have potential for being used in some form of therapy to replace tissues or organs which have been damaged by disease or accident. There's obviously tremendous advantages to possibly non-reproductive cloning. So the cells will be identical to host cells, therefore no rejection. There wouldn't be issues with donor organ shortage. Cloned totipotent cells, we'll come back to what totipotent means in a minute, can differentiate into any cell type, which is quite exciting. And using cloned cells can avoid major invasive surgery, which particularly in this day and age with various MRSAs and C. difficiles, we try to avoid invasive surgery as much as we can. And then there are various possibilities, and you can look these up yourself, but there's all sorts of things. One that's talked about a lot is the idea of putting cloned stem tissue of some sort into damaged heart tissue after a heart attack, and that will regenerate the heart tissue and bring back the function of the piece of heart that died in the heart attack. And then also talked about repair of nervous tissue following disease or after some sort of accident, broken neck type injuries. If we could grow spinal cord at the area of, grow new nervous tissue at the area of damage on the spinal cord, it's possible that we might be able to return some function to the patient. Okay, so that's ideas of reproductive and non-reproductive cloning, which you have to be able to understand. Little diagram which I think I had on my previous slide, which I'll just quickly go through again. So this shows you nuclear transfer, which we'll come back to and talk to you about in a second. So we've got the cell which is going to donate our nucleus. Okay, we've also got an egg cell from the same species. We take the nucleus of the egg cell out, so we've got an empty egg cell, which is called an anucleated egg cell. We then take the nucleus out of our somatic cell. We then place the nucleus into our egg cell. We allow that egg cell to develop into a ball of cells. Okay. Now, what we do with those ball of cells determines whether we're talking about reproductive cloning or non-reproductive cloning, which is sometimes known as therapeutic cloning. So reproductive cloning is that ball of cells is then grown, okay, and then placed into a surrogate mother, and then that develops into an individual. Okay, so that's, for example, Dolly the sheep, the much vaunted Dolly the sheep, was produced in that way. Therapeutic cloning takes this ball of cells, okay, and then use these individual cells for other purposes, okay, for research or for some form of some sort of therapy, which might be useful. Okay, so we're not actually making individuals; we're using these cells for some sort of scientific research or therapy. Okay, so that deals with the principles behind cloning. We move to the next slide. This slide deals with how artificial clones of animals can be produced. Okay, so it all is based on the idea that totipotent stem cells can differentiate into any type of adult cell. Okay, which again, the word totipotent is something you will have come across in your F215 course. So a totipotent cell is one of these stem cells which can divide and differentiate into any other type of cell. Okay, so they then divide into what we call pluripotent cells. These can only divide into a more limited number, and then other stem cells will be able to divide into an even more limited number. But totipotent cells are ones that can divide into any type of cell eventually. Okay, so that's a key word, totipotent stem cell. And talking about different ways in which we can do this. The first thing is embryo splitting, and this has been around for a long time, and it involves taking a newly fertilized embryo and then separating that ball of cells into individual cells and then growing them in a laboratory until they're a sufficient size, when we can then insert them into surrogate mothers who then produce offspring. Okay, so here we have a cluster of identical cells, okay, which have been produced by a recent fertilization. Okay, so this would be, let's say, of a cow with a particularly high yield, milk yield that we're interested in. Okay, and we've got these cells which are been recently fertilized. We then separate those cells into individual cells. We then grow these cells in the laboratory until they grow to a certain size, a size when they can be reimplanted into these different surrogate cows. Okay, and these surrogate cows will all produce cows which are the same. Okay, and hopefully these cows will all have a large milk yield. Okay, so that's embryo splitting is one way that we can clone animals in that way. The next way is called nuclear transfer, okay, and we talked about this already. And what we do is we take the adult nucleus, okay, is fused with an anucleated egg cell from another adult, okay, and then electrofusion occurs, which stimulates those cells to replicate, and then once the ball of cells are sufficiently large enough, it can be inserted into the uterus of a surrogate mother, and then a clone of the adult from which the original nucleus came from is produced. Okay, as I say, the famous Dolly the sheep, which was produced some time ago now, was produced in this way. Okay, representing all that diagrammatically. Now here is Dolly the sheep. Okay, so I have a sheep here, okay, which has donated an egg cell, which I've removed the nucleus from. Okay, that's called anucleation. Okay, to produce an anucleated egg cell. Okay, I then took a somatic cell, which in this case was a mammary cell from a Finn Dorset type of sheep. Okay, and I fused the nucleus, or fused this cell with my anucleated egg cell. Okay, I've then given it a direct current pulse, okay, which stimulates it to divide, okay, and it divides into an embryo. Okay, this is a part of embryonic development, and that embryo can then be implanted into a surrogate ewe, okay, and then however many months later a sheep was produced. This was Dolly, and the idea is Dolly is a clone of this Finn Dorset type sheep here. Okay, it's got the same features as this type of sheep here. Okay, so that's nuclear transfer. Okay, a nice little animation of that happening below. It's very simple, which you can watch. So here is our anucleation. We take the nucleus out of our egg cell. Okay, that's the nucleus. So this is an anucleated egg cell. We then take the nucleus out of our somatic cell, which is just a skin cell, and then we put that nucleus into the egg cell. Okay, and then the idea is that this will then grow and divide. Okay, it's usually given a pulse of electric shock at this stage. Okay, so it will then divide by mitosis. Here we go. So it's divided into 2 cells, 4 cells, and eventually divides into a ball of cells, which then divides into an embryo, which can then be implanted into the uterus. Okay, so this is it can be implanted into the uterus of some sort of surrogate mother. So moving, this very simply goes through some of the advantages and disadvantages of animal cloning. Some of the advantages would be that we can get large numbers of animals which have got a desired characteristic. Okay, so we talked about high milk yield. It could be the cattle which grow particularly quickly, or it could be salmon which have got a high growth rate. It could be all sorts of things we could be looking at here. Okay, it can also be useful in conservation. Okay, cloning rare animals can quickly increase the population of those animals, which can be very helpful in conservation. And also genetically modified animals can be rapidly reproduced. Okay, so you could genetically modify an animal to have some form of advantage. Okay, and then we could use, say, embryo splitting to produce lots and lots of those. Okay, disadvantages. Okay, animal welfare often ignored. Okay, so high meat yield chickens are so big they can't walk. Okay, so breeding has produced these ones that can't walk. Okay, so that's not particularly good for animal welfare. And also the genetic uniformity would prevent adaptation to any environmental change. And therefore if a disease came along it could wipe out all of your population. And also we're unclear about the long-term health effect of clones produced by nuclear transfer. Okay, it's not as straightforward as it seems, in that the products of these nuclear transfers seem to develop problems later in life. Okay, so those are some of the advantages and disadvantages. So that concludes my screencast on cloning in animals.